Thanks for being back. Excited to get to this one. This is Beken Bio, and it is an early stage biotech startup and developer of liquid biopsy-based cancer detection technology intended to develop minimally invasive diagnostic tests. The company's engine aims to transform cancer care by identifying traces of tumor communication found in patient blood in the form of cancer-specific proteins transported by EVs, which are extracellular vesicles, exosomes, right? Thereby exposing cancer, specific, again, these extracellular vesicles that circulate in the blood earlier and in significantly greater quantities than other liquid biopsy targets, which enables clients to save lives and explore new horizons of nanobiology in conjunction with state-of-the-art machine learning AI. I actually had Paul Billings on one of my podcasts, and the way they describe it is, you have pheromones and you have this kind of like must or sweat that you can tell of the individual without actually biopsying. And there's a lot of that communication that goes on between cells. So this is something that's super exciting to hear that y'all are working on, because Chris Millan, who is our CEO of Beken Bio, grew up in New Orleans from across the river here. He studied biomedical engineering at Tulane and then went abroad to the Swiss Institute of Technology in Zurich for graduate school. There, Chris worked on engineering 3D materials to grow cells in the labs as if they are still in the body, and was recognized on the annual Forbes 30 Under 30 list in 2017 for his contributions to the field. During his postdoc, he developed the idea to use engineered tissue models to study their secreted factors as a means of biomarker discovery, and the idea for Beken Bio was born. The company is currently in its pre-seed stage and is based in San Diego Biolabs, which plans to move operations home to New Orleans later this year as part of their commercialization plan for their first product, an ovarian cancer diagnostic. Thank you.
Thanks for the introduction, and I just want to start out by saying thanks to the CAGLA folks, especially Chad, for this opportunity. I was a kind of late fill-in, and it's a wonderful opportunity for us. I'm sorry for being the last company to get our presentation in. Hope it didn't stress you out too much with that. So I'm here to tell you about Beken Bio. As we covered a little bit in the intro, we're obsessed with these cell messengers called extracellular vesicles. And they don't look like much, but we have 35 trillion of these zooming around in our bodies at any given time. And basically, cells use them to package molecular cargo like proteins and DNA. They send them out into the blood to deliver instructions to cells located somewhere else in the body and get them to change their behaviors. So if we can decode this cargo that are present in these EVs, we can figure out what kind of cell made them. And cancer cells use these to a great extent. They secrete a large number of them, and as this primary tumor is growing, they're sending these EVs out with the goal of preparing the eventual metastatic site to get it ready for when that first cancer cell breaks off the tumor and latches on. They're present earlier and in much higher amounts than any other cancer cell-specific marker. That includes DNA and CTCs. And so they're wonderful targets for highly sensitive early-stage tests. And I know we're all aware that early detection is very important. The statistics are very scary. 39% of us will be diagnosed with cancer at some point in our lives. And the point or the timing of detection is one of the most important factors that determines the survival. And as we heard from the previous talk, ovarian cancer is one of these indications that the majority of the patients are diagnosed late. And that's just because of where the ovaries are and that it's largely asymptomatic in its early stages. So if we have tools that can pull these EVs out of the blood and quantify the specific markers in them that correlate to proteins for very particular cancer indications, we can potentially screen patients and shift the probability for indications like this to earlier detection. But it's not as easy as that first video that I showed you seemed to indicate. When you actually look in the plasma, patient plasma, EVs are outnumbered by up to 8 orders of magnitude by these other particles, lipoproteins, cholesterols, things like that. And that makes it very difficult to quantify them. So there's some technical challenges that have kind of been roadblocks for getting EVs onto and into the clinic. The two challenges that are faced are just being able to see the cancer EVs through all of these other contaminating things in there, and then getting a pure enough population of EVs to then figure out what that fingerprint, that molecular fingerprint is that would correlate a specific EV to a specific type of cancer. And at Beken, we've developed a suite of technologies to overcome those challenges. And together they comprise these novel diagnostic tests that allow us to quantify EVs from patients. The first tool is for purification of EVs from plasma. And basically, if you plot these different particles that are present in the plasma, EVs separate out based on size and on surface charge. And so what our team has done is built a special chromatography device that does dual mode separation. Basically, you put the plasma in, and there's a composite resin that separates everything that's too small out and everything that has this negative charge out, so that our positively charged large EVs flow through, and we're left with a very pure population of EVs to quantify downstream. Here's what it looks like in practice. It's just basically a syringe. You can see the composite resin there with a small bit of patient plasma on top. And this technology was developed by one of our team members during his postdoc at Harvard Med School. And we have an exclusive option agreement in place for licensing that patent. Our second technology is for pinpointing the molecular makeup of EVs from specific cancer types. And this comes from my own postdoc work in Zurich, Switzerland. As was mentioned, I was working on engineering microenvironments to grow cells from the body in the lab, but have them still behave as if they're still in the body. And what that looks like this is a picture here of primary ovarian cancer cells grown in one of these 3D environments. And they form these beautiful 3D networks, but more importantly, they express tumor antigens in an enhanced rate. So there's a higher abundance of cancer-specific markers when you grow cells this way. And we figured out that also translates into the EVs that those cells make. So what we do is we put primary cancer cells in this system, we take the EVs that they make, and we study them via a label method of proteomics, mass spectrometry. And that basically tells us, we figure out the high abundance rate of certain proteins, and that gives us the key to then engineer a diagnostic test for a given cancer type. Here's some data from our first run, which was for ovarian cancer. We partnered with the Cornell Medical School to source primary ovarian cancer cells. And we did the proteomics on the EVs here in New Orleans with LSU. And I know the data looks a little complicated, but I'll walk you through it. This is just a heat map of a panel of 20 proteins. Each column represents one patient, and each row is one protein that we looked at in terms of the abundance that those EVs from that patient had. So you can appreciate that the first few, which are the healthy ovarian cells, have very low abundance. So these blue squares represent low abundance for those proteins. And then for each of these subtypes of ovarian cancer, you see that there's a very specific signature. So ovarian cancer is not one disease. It's actually a very heterogeneous disease, and that's really important. I'm going to come back to it, but basically the treatment for each type is very different as well. And so it's important that we can kind of differentiate between them. And so this basically allows us to know which proteins are cancer specific and build our tests accordingly. And finally, we use machine learning to evaluate a list of proteins like that panel I just showed. And basically what our machine learning algorithm does, it looks at different patterns from patient samples and allows us to build a classifier that can then decide whether this patient has cancer or not, or likelihood of cancer or not. So we develop clinical tests. This is kind of a platform technology that we can apply to many different cancer types. I mentioned that we chose to work with ovarian first. But how do we plan on commercializing the test? Well, there are certain companies that have had a lot of success, and we're just following blueprints that are out there. But we have a three-pronged commercialization pathway here. The first is to bring the tests to market as laboratory-developed tests, or LDTs. And to do that, there are a number of advantages. The regulatory hurdles are a little bit lower, so you can get onto market faster as an LDT, but you have to offer that test as the provider in-house. So the samples have to be taken at point of care, sent to your lab where you process them, and you provide the data back to the clinician that can then use that for, it's actionable information for them. And the way it is regulated is by a series of basically standards called CLIA, that look at validation data for diagnostic tests, as well as how the facilities are set up and how the facilities are run. So they look at things like how you do instrument maintenance, how you track a patient sample when it gets to you, goes through your process, comes out, and making sure the data gets back coded to the right patient, things like that. We plan to come online with our ovarian cancer LDT by the beginning of next year. And we want to do that here in New Orleans. We've already been in talks with the team at the New Orleans Bioinnovation Center, which is just, I don't know where we are, just up the street from here towards the highway. And our tests, or the indications that we'll work with soon after that, are lung cancer, followed quickly by colorectal and pancreatic, which have different markets and different prevalence rates. But all of them have this thing in common where the majority of the patients are diagnosed late, and we hope to change that. We will first partner with local cancer centers, and we've been in talks with the relevant people at Mary Bird Perkins and at LSU to do our first validation, with real prospective studies in these clinics. They would be our early adopters in this case. And then from there, we would step towards working with, there are 15 comprehensive cancer centers in the country that have high-risk ovarian as a specific branch of their departments. And we would look to use them as early adopters as well. They would have clear incentive to do that. And in parallel, this is something that was talked about this morning by Amir Ali as well. It's very important that you work very early on with payers. And our idea is to do pilot studies with Blue Cross Blue Shield and others to offer our test to a subset of their patients as a way for them to evaluate the clinical value as well as the financial value of covering a test like this so that more people can have access to it. The second commercialization arm puts us in between pharmaceutical companies and the FDA in their clinical trials. And I mentioned that our test seems to have the ability to distinguish between subpopulations of cancer types. And like for ovarian, clear cell ovarian cancer doesn't respond well to chemotherapeutics. And if you look in the clinical studies database, there are currently 170 clinical trials right now trying to pair immunotherapeutics to specific treatment for clear cell ovarian carcinoma. And we would be able to help pharmaceutical companies find patients for those clinical trials with our test. And in this case, it's a research use only. So there is no, we could start selling that test tomorrow. Of course, you need to publish. You have to get the data out there. And then the way you basically build these relationships are at these very large partnering meetings. There are these conferences like BIO where tens of thousands of members from the pharmaceutical community attend, and you can set up one-on-one meetings. I've been to them before. It's a marathon. But it's a wonderful opportunity to find those connections. And finally, I've also been in touch with some of my colleagues. I spent 10 years in Switzerland. I did work at Roche, collaborated with Novartis, and have some friends in those spaces that might be willing to give us a shot in this regard. And then finally, we would like to sell the test ultimately as in vitro diagnostic devices. And in this case, we would sell the kit that would basically include that column for doing the EV purification, as well as all the reagents to quantify a specific panel, for example, for ovarian cancer. And we would sell that to a company like Quest or LabCorp to run the test. And this is obviously a much more scalable model for us, but it does require FDA approval. So we would leverage the data that we generate during the LDT phase to apply for that 510K designation with the FDA. And ultimately, we would, when the timing and the data supports it and is appropriate, we would appeal to the U.S. Preventive Services Task Force for recommendation, again, for basically using our test as a screening tool for these indications. First, for lung cancer, because it's far more prevalent, so it's easier to demonstrate that it has utility as a screening tool, and the others would follow shortly after. Now, looking at the market, there's been a recent analysis just from last year looking at the global market for blood-based liquid biopsies for cancer specifically, and it's pegged the market size at $4.18 billion. The US represents about half of that market. But for our first obtainable market, we're looking very specifically at the two largest payers here in Louisiana. That's Blue Cross and United Health. This would be kind of a market projection for our first couple of years online. I think what speaks more to the potential growth of the market is just the amount of money that has been invested in this space, either from acquisitions or straight investments. And you can see that we differentiate from a lot of companies in the fact that we are the only one that's doing both EVs for our diagnostic tests and doing bottom-up biomarker discovery for novel biomarkers. So the fact that we do EVs gives us a lot of confidence that our tests will be very sensitive for early stage. And the fact that we're doing bottom-up biomarker discovery gives us confidence in the specificity of our tests. And I want to highlight a few because there are a couple out there that have looked at ovarian cancer and we can compare one-to-one with them. So I'm going to show you data that shows how we stack up against these other companies. But first, I want to show in a more granular detail the data from our preliminary clinical validation work. We've had the opportunity to test 100 patients so far, including control, early stage, and late stage. And a few of our markers have shown very high statistical significance between the controls and the cancer patients. And more importantly, the AI-based algorithm has come up with a 97% probability rate. That's the AUC on our ROC curve here for its ability to classify patients correctly. And if you compare with the same, for the same patient group, this CA125 that the previous presenter mentioned, you can see that there's a correlation for increasing levels of this marker in the blood with later stage cancers, but it's not statistically significant and the AUC is quite low. As I mentioned, just want to show these data, and I don't mean to pick on these companies, it's just specifically they're looking at DNA, we're looking at EVs in some cases, and their tests are wonderful for multi-cancer detection, but maybe not great for early stage of a very specific indication. So the sensitivity values for the two EV tests here are high. That's because EVs are present or in earlier stages than DNA. The specificity levels are high for all of these tests, as well as the NPVs is more because ovarian cancer is just a little bit less prevalent than other cancer types. But I want to highlight the positive predictive value. And it seems like a low number, but the threshold that's kind of been discussed in the gynecological community is 10% for a test to have utility as a screening tool. And as far as we know, we're the first to reach that mark with this test here in our small preliminary cohort. Now I want to talk about the funding situation. So far, we've raised $500,000 through angel investments and non-dilutive prizes. And we've invested that in setting up our lab in San Diego and in development of this ovarian cancer test. And where we currently stand, we want to finish this pre-seed round with another $600,000. This would get us onto the market and ready for launch of the ovarian test as an LDT. We would hire 2 additional people, apply for this CLIA certification, and be able to come online in early 2025. This would also allow us to reinvest in our pipeline and expand it to first lung cancer and then soon after colorectal and pancreatic cancers. And very soon afterwards, we'll need to raise a larger seed round to then basically fund hiring of regulatory people and salespeople and focus on growing our customer base in this sense, as well as growing our portfolio, again, targeting lung cancer as the next test to come online there. Finally, I'd just like to highlight our key team members. You've heard about me. I had the chance just before COVID to do this Mass Challenge Accelerator program, and we were fortunate to win some non-dilutive funding there. Devin was my mentor during that process and has been with us as an independent director since founding. He's brought LDTs to market, gotten CLIA certification for labs, and even has had a couple of exits to Roche and Novartis. Jan is the scientist that I mentioned. He was the founding member of our scientific advisory board. He invented the DMC technology. And then we have 2 brilliant bioinformaticians from Accelera that we work with for the machine learning portions of our R&D. And clinically, I mentioned already the relationships we have with Cornell for the primary cancer samples, and then the 2 clinical validation networks that we have with LSU and Mary Bird Perkins. So with that, I'd like to conclude and just say that Beken Bio is excited about unlocking the potential of EVs for diagnostics, and we've made significant progress to date, and we're at a very pivotal moment here looking for any collaborators, investors, anybody with questions or feedback, very willing to listen, and thank you so much for your time. Thank you. Yes.
So, Chris, you said you've got 500,000 so far. You've raised 500,000. You only need another 600,000 to come out with a commercial LDT product. That 600,000 will get you through FDA.
So it's not FDA, but it's just applying to the local state's Department of Health, and it will basically allow us to set up the lab and have everything online for that application, for the CLIA certificate, which is all.
The 600,000 won't get you a product ready.
it will get us ready for that, just the LDT, not the in vitro diagnostic. They're very different. The in vitro diagnostic, I think the application alone is like 50,000 to the FDA, and you need a wealth of data. So we're a long way from that.
But the LDT is a product that you can take samples, do the test, and give results.
Right. Correct. Correct.
And that doesn't need FDA.
Nope, it does not.
Yeah, well, on that topic, I saw something recently in the New England Journal of Medicine that FDA wants to reevaluate this whole process. They think
Yes. 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 Yeah. It's a little bit shocking, but there is some, I mean, you still have to apply to the Department of Health. There is still a control that the test does what you say it does. And there has been a lot of discussion for, I don't know, 20 years at least. And so we're obviously monitoring it, and that's why we have this idea to go from LDT to in vitro diagnostic. That's where we want to get, but it's just a wonderful opportunity to get on the market and build relationships with clinicians, even if it's just locally, and just kind of get some traction before putting that application together.
The FDA's oversight on this, potential oversight on this, they're not looking until 2028 now.
Yeah. Yeah.
There's thousands of tests that this could take them forever.
Very likely. Exactly. And so we're kind of, I've spoken with some consultants about it, and it looks like, if we make it in the next year or so, we'll be grandfathered in under these old stipulations. Yes. Thank you. Mm-hmm.
for some time. That being said, one comment I would make is that if you don't have a clean lab set up already, often it takes about a year to
Mm-hmm.
just get there.
Mm-hmm.
Whatever your financials.
It's surprising that you just have to apply for it, and as soon as you apply for it, you can start selling the test. Yes. Yeah. So I want to do it here, and I've already been in touch with the people in Baton Rouge that run CLIA for the state of Louisiana. I think it is. Yeah, I think it is. Please. Mm-hmm. Correct. Correct. Right, but usually the composition of the outer membrane of the EVs is the same as the cell. And so you have the glycocalyx that is positively charged overall. And therefore the surface, if you look at just zeta potentials, EVs are very highly positively charged compared to lipoproteins.
I missed the very beginning, so on that point.
Yeah. We have to be very careful. I mean, if you attend a conference on EVs, they're very strict about what you call an exosome. So EV is kind of like this blanket term that covers exosomes, microvesicles, a lot of different things. And to be an exosome, there's a very strict criteria that you have to go through. So just to be careful, we just say EVs, it's this blanket thing, and we don't necessarily care if it's a canonical exosome, which is a very specific biological pathway and how it's made and things like that. Yeah. Thank you guys so much.